pentose phosphate pathway is a pathway where we are going to be able to form 5-carbon phosphates, which will give rise to other stable intermediates that can be used in many other pathways. We start out with glucose 6-phosphate. This is the same material that you're going to have when you deal with glycolysis. First step is to form glucose 6-phosphate. From here it says uses an NADP, form an NADPH, and the enzyme is glucose 6-phosphate dehydrogenase. That is going to give you 6-phosphoglucanolactone. You can see how it seems to have a rather unique format in here, and what has happened is the first and the 5th carbons are actually bound together with an oxygen in the middle, giving it kind of a ring-shaped structure. From here, the enzyme lactonase comes in. It says it takes a water, releases a hydrogen from the water, and we're going to get 6-phosphogluconic acid, which is a little bit different from the glucose 6-phosphate because it now has an acid group up on the end. Then an enzyme called 6-phosphogluconic acid dehydrogenase comes in. It again uses an NADP, which is going to produce an NADPH, and it's going to evolve off of carbon dioxide. From that, we are going to get the formation of ribulose 5-phosphate. Ribulose 5-phosphate is now a 5-carbon base. This 5-carbon base, with an enzyme called ribose 5-phosphate isomerase, will form ribose 5-phosphate. Notice the word ribose in there means that we are looking at building blocks for RNA and DNA. You've got ribose 5-phosphate. There's another enzyme that's called phosphopentose epimerase that will cause the formation of xylulose 5-phosphate. And again, it's just changing the position of some of the oxygens within this unit. The unique thing here is there's an enzyme called transketolase. This is going to take the ribose 5-phosphate and the xylulose 5-phosphate, use them to create other things from it. So you take 2 5s, and out of that it makes cetoheptalose 7-phosphate, which is a 7-carbon unit, and glyceraldehyde 3-phosphate. Now that's another name that you're going to get familiar with because glyceraldehyde 3-phosphate comes not only out of glycolysis, but you also find it in the Calvin cycle. Another transaldolase can take these 2 units, the 7 and the 3, and form fructose 6-phosphate. Again, we're back into the glycolysis, and erythrose 4-phosphate. Now, the erythrose, again being a 4-carbon unit, can be used for the making of other things such as amino acids. Another transketolase can start out with the xylulose 5-phosphate and the erythrose 4-phosphate, that's a 5 and a 4, and out of that give you glyceraldehyde 3-phosphate and fructose 6-phosphate. So we're creating units where we shift units around. 2 5s make a 7 and a 3, a 5 and a 4 make a 6 and a 3. So we've got many different possibilities out of this, and what we get out of all of these is all of these are phosphates, which means they're still reactive and they can be used for other materials. That's what we call the pentose phosphate pathway, because one of the first products we create is a 5-carbon or a pentose phosphate.